Hi everybody, happy Jurassic June. I'm David Button from the University of Bristol, and I'm here today to talk to you a little bit about the ways in which we reconstruct the lives of extinct dinosaurs. Now, as we all know, the Jurassic Park franchise has been responsible for some of the most iconic depictions of dinosaurs in pop culture, to varying degrees of accuracy. It is also responsible for some of the most iconic depictions of paleontology and paleontologists as well, although I can only wish it was quite as easy as finding an entire velociraptor skeleton just buried under some sand, and I can certainly only wish that I could be quite as dynamic as Drs. Sattler or Grant. But nonetheless, not a great deal of the paleontological process is depicted in Jurassic Park or the subsequent movies, and that's something which I'm hoping instead to bring to you a little bit today. Now, as we all know, in the Jurassic Park franchise, the recipe for dinosaurs goes something like preserved mosquito and amber, plus scientists, plus sophisticated techniques, plus DNA from a frog equals occasionally misspelled dinosaur genome. Now, about the time that Michael Crichton was writing the original Jurassic Park, there was a great buzz in the scientific community about claims of potential preserved DNA being intact in Mesozoic era fossils. Unfortunately, however, subsequent examination showed that these early supposed cases of Mesozoic era DNA were instead the result of more modern contamination, and so sadly, no dino DNA. And although more modern claims do exist, it is very difficult to separate any such results from genetic material that will be present from microbes also inhabiting the fossils themselves. And indeed, the oldest, broadly agreed upon, well-supported genetic material that's extracted from fossil remains come from some 1.2 million year old mammoth teeth, as reported upon earlier this year. So not a dinosaur, but still pretty cool. Nonetheless, it really does seem in this case that we're starting to push the boundaries for how long significant amounts of genetic material can survive in the fossil record. And though a 1.2 million year old mammoth tooth may sound old, there's still a massive gulf between that and a 70 million year old dinosaur fossil. And so sadly, I for one believe that the half-life of DNA is just too short for significant amounts of genetic material to ever be preserved in any Mesozoic era fossils. So yeah, sorry about that, guys. Nonetheless, we are starting to discover that exceptional preservation may be far more common in the fossil record than we once thought, with purported cases from dinosaurs including the preservation of fragments of proteins and amino acids, and even the replication of the shape of dinosaur blood cells. And taken together, these kinds of discoveries could teach us a lot about dinosaur biology. So, as this lack of DNA leaves us sadly unable to clone dinosaurs, even with the help of an eccentric billionaire, then how exactly are we supposed to reconstruct their appearance? Now, in some cases we're exceptionally lucky, and exceptional preservation provides a window through which we can reconstruct dinosaurs with a great degree of confidence. Good example of this are the well-known hadrosaur mummies, which show copious skin impressions and in some cases even preservation of organic material that shows us that these animals were covered in a pebbly mosaic of scales. And indeed, soft tissue preservation of Edmontosaurus shows us that this dinosaur had a soft tissue comb on its head, a bit like a chicken, and further showing us that even what we thought was one of the best known dinosaurs still had a few surprises in store for us. And of course, in recent decades, these examples have been joined by large numbers of specimens, particularly from China, of dinosaurs preserving the presence of feathers, something which has completely overhauled our understanding of their biology. And this has included such treats as the crow-sized dromaeosaur Microraptor, which sported wings on all four of its limbs, of the velociraptor-sized dromaeosaur Zhenguanlong, showing us that raptors too would have been fully decked out in a plumage of feathers, and most surprisingly of all, the small theropod Yiqi, which shows not only feathers on its body, but also bat-like wings of skin on its arms, preserving a form of flight that was completely unexpected in the fossil record. And even if we look beyond theropods, we see in ornithischians such as Psittacosaurus the preservation of quill-like structures which may or may not have anything to do with feathers. Remarkably, some of these feathers show subcellular levels of preservation, and so when placed under a scanning electron microscope, we can see the melanosomes, or pigment organelles, within them. Now, although it is important to remember that there are many forms through which feathers are pigmented, not all of which preserve in fossils, comparing the shape of these melanosomes to those of living birds does allow us to start to reconstruct some of the colours that these fossil dinosaurs would have been. So, for example, Anchiornis seems to have had piebald body feathers and a rufous crest, whereas Microraptor had iridescent feathers like those of a crow. Now, in light of these amazing discoveries, it remains perhaps inevitable that the main concern of the media often seems to be, does this mean Tyrannosaurus rex had feathers? Well, this is an argument that got a real shot in the arm with the discovery of Eutyrannus, an earlier tyrannosauroid which sported a pelage of filament-like feathers. Now, Eutyrannus was about 9 meters long and weighed around a ton, and so this proves that at least some large-bodied tyrannosauroids did indeed have feathers. But what about the tyrannosaurids proper, like Albertosaurus and Tyrannosaurus rex itself? They, after all, got a lot larger than Eutyrannus. Well, more recently reported skin impressions from Tyrannosaurus rex show pebbly mosaic-like scales from across large parts of the body, including the neck, the lower back, and the tail. And further, some have suggested that textures on the facial bones of Tyrannosaurids such as Daspletosaurus suggest that it was armoured with scales. Now, taken together, these then suggest that large tyrannosaurids like Tyrannosaurus were at least primarily scaly, although it should be noted that we do not have complete coverage of the body, so the presence of some filaments on areas such as the upper back cannot be entirely ruled out. And further, as well as soft tissue preserved around dinosaur bones, if you're very lucky and ask very nicely, and someone is kind enough to let you cut through a dinosaur bone, you can also find a lot of information preserved within it. So one example here is the cited presence of medullary bone in the bones of some dinosaurs. Now, medullary bone is a tissue that's laid down by living birds when they're in the process of forming and laying eggs, as it helps to protect the calcium from being leached out of the mother's bones by the developing eggs. Consequently, if we could find medullary bone in prehistoric dinosaurs, this would be very exciting, as it would allow us to establish which specimens were then adult females in the process too of egg laying. Unfortunately, however, it should be noted that the distribution and form of medullary bone is actually quite poorly characterized among living birds, so many cited instances of medullary bone in fossil dinosaurs should still be treated with some degree of caution for now. Now, look at the texture of dinosaur bones, we can also see growth rings in many cases, similar to the growth rings of a tree. And in the same way, if we count these growth rings, it allows us to establish the age of the dinosaur when it died. Now, if we have large numbers of dinosaur specimens of a particular species, this then allows us to establish growth curves by figuring out how large a dinosaur was at various ages. And particularly interesting, if we look at the fossil record of hadrosaurs and tyrannosaurids, we can see both that these animals were growing very, very quickly, but also there seems to have been something of an evolutionary arms race between them, with the duckbills racing to grow to reach fighting weight as quickly as possible, and the tyrannosaurids in turn trying to grow as quickly as possible to keep pace with them. Now, what is interesting is that we see these very high growth rates in dinosaurs. And these are more similar to those of living warm-blooded mammals and birds than they are to living cold-blooded reptiles. And so if we synthesize these data, both from growth rings but also from the overall texture of dinosaur bones, then this suggests that they too showed elevated metabolisms, and indeed we can see the parallel evolution of warm-bloodedness both within the dinosaur lineage that led to birds and also in the synapsid lineage that led to living mammals. And so this very much leads us to this concept that dinosaurs were not like modern ectothermic cold-blooded reptiles as once thought, and indeed many of them, particularly smaller ones, would have been more similar to modern endotherms, such as mammals and their extant relatives, the birds. However, it should be noted we're increasingly beginning to understand that metabolism is not a simple binary between ectothermic and endothermic individuals. And in particular, much attention needs to be given to mass homeothermy. That is, even large-bodied reptiles, like leatherback turtles, are able to maintain a stable core temperature, in part due to their large body size. And in particular, it should be noted that the large body size of many dinosaurs means that effectively they would have been able to maintain a stable temperature, whether they were endothermic or ectothermic. Consequently, it seems best to think of dinosaurs as ranging across the spectrum we see in living animals. So whereas they all seem likely to have had metabolisms elevated above those of modern ectothermic reptiles, many of them still probably had metabolisms below the level of modern birds and mammals. Now, you are what you eat, and the diet of dinosaurs left many traces on their skeletons, most particularly on the teeth. So if you subject dinosaur teeth to high-powered magnification under a microscope, you'll see the presence of minute scratches and other microwear textures that not only help us to narrow down what a dinosaur ate, but also show us how the tooth rows move past each other and so reconstruct the jaw motions that these animals used to procure and process their food. So for example, analysis of the tooth microwear textures of the small ceratopsian Leptoceratops show that it combined vertical motions of the jaw with forward and backward sliding in an orbital bite cycle reminiscent of that of living rodents, that in a similar way would have allowed Leptoceratops to effectively slice its food. Similarly, analysis of the microwear textures of ceratopsids show that they too likely performed some degree of backwards and forwards movements of the lower jaw, but also that they performed very powerful vertical shearing driven by the strong jaw muscles these dinosaurs had. Taken together, these show that ceratopsids clearly had a very powerful feeding apparatus that would have allowed them to effectively slice through even the toughest plant material, and consequently, ceratopsids do not seem to have needed or used gastroliths, which is of course great news if you wish to house them alongside poisonous berries. The real champions of dinosaur teeth, of course, were the hadrosaurs such as Parasaurolophus, which combined hundreds of small teeth into powerful tooth batteries of which to grind their food. And recent work exploring the histology of these batteries is showing that how they were a dynamic, reactive structure, providing an extremely complex surface over which these animals could grind their food with unparalleled efficiency. So we can see that a lot of information is preserved in fossil dinosaur bones as well as soft tissues, but how do we reconstruct soft tissues when they're not there? Well, something which has been a great aid to this has been the increasing use of CT scanning in dinosaur research. And this allows us to take dinosaur specimens into the virtual environment, where we can perform various retrodeformation procedures to correct the damage that was done to them during the fossilization process. So we can fill in holes, repair cracks, and as well as undo the warpage that the bones will have suffered while being compressed by sediment. And so this allows us to reconstruct parts of the body, such as the skull, as they would have been when the animal was alive. And this 3D reconstruction is very useful. We can look at it in all angles and we can readily share it with our colleagues. And we can also use the CT scans to peer inside the bones to look at, for example, the replacement teeth growing in the skull and work out how quickly they were replaced. Finally, you can use scars and other features on the bones with comparison to living relatives like birds and crocodiles to reconstruct soft tissues such as the muscles, for example, the muscles of the jaw, and to figure out their orientations and their relative sizes, which gives us an idea of how strong they would have been able to contract. And so by piecing these forms of evidence together, we can really start to get a comprehensive look at what dinosaurs would have been like in life. Now, a particularly interesting interior space of the skull to reconstruct is of the interior surface of the brain case, which allows us to look at dinosaur sensory biology. So, for example, reconstruction of the brain of Tyrannosaurus shows a well-developed olfactory tract, indicating a powerful sense of smell. Similarly, reconstruction of the inner ear of Velociraptor indicates not only that it had an effective sense of balance, but also that its hearing was finely tuned to the high-pitched squeaks of its small mammalian prey. Now, looking at these things in the Alvarezsaurid Shuvia has shown even more remarkable results, indicating that a highly elongate cochlea, similar to that of the living barn owl, which would have granted it similar auditory acuity. Similarly, we can also see from the dimensions of its sclerotic rings that it had very large eyeballs, similar to living nocturnal birds, and taken together, this shows that an extremely advanced nocturnal habit was already present in this Mesozoic dinosaur. And this also allows us to estimate the volume of the brain, and this is interesting because the size of the brain relative to the body, as well as examination of its various substructures, provides a very rough estimate of intelligence between different groups of animals. And so here we have a plot of the relative brain size of various groups of archosaurs, and we can see that unsurprisingly the sauropods fall out at the bottom, although they do still fall within the range seen in living reptiles. By contrast, many other groups of dinosaurs show relatively larger brains, more similar to those of living birds than reptiles, and in particular, the small theropod groups such as Troodontids and Dromaeosaurids come out on top. They shouldn't look too smug, however, because if we compare them within the broader range of brains as seen within living birds, we can see that non-avian theropods do not cluster with the relatively large-brained living birds like crows, but instead they show greater similarity to more basal land birds. So perhaps we shouldn't be thinking of raptors as smart like crows or parrots, let alone apes, whatever Jurassic Park 3 says, but we should instead be thinking of raptors as being smart like pigeons. Now, this may not be as bad as it sounds. Pigeons are actually very adept at memorizing complex spatial and other forms of information, and it should also be remembered that the relative size of the brain is only a very rough estimate of intelligence and may be of dubious utility, particularly when being compared between animals of very different sizes and lifestyles such as these. But nonetheless, what this does show us is that dinosaurs would have been capable of learning complex behaviours. And what these reconstruction techniques also allow us to do is to consider dinosaurs mathematically as biomechanical problems by looking at how the reconstructed jaw forces would have moved components of the skeleton to perform work. So, for example, a lot of attention has gone into computational biomechanical modelling to calculate bite forces in Tyrannosaurus rex, because this sort of thing is always done on Tyrannosaurus rex. But the results are actually very interesting, and they show that calculated bite forces in Tyrannosaurus vastly eclipse those measured or calculated for any other land animal, much greater than even those of the saltwater crocodile. And this would have granted Tyrannosaurus with the bite power it needed to inflict the deep puncture marks and even entire removed sections of bone observed in ceratopsid and hadrosaurid fossilized bones that were fed upon by tyrannosaurs. It also would, of course, granted Tyrannosaurus all the power it needed to break through as many car roofs and ingest as many lawyers as its heart could possibly have desired. Interestingly, if we look at some calculated bite forces for some other large theropods, we see that these are much lower and, oddly, are similar to or even lower than crocodilians or even mammals of much smaller body sizes. So what is going on here? Some authors have investigated this problem using other techniques, such as finite element analysis. Finite element analysis is a biomechanical modelling technique which allows us to reconstruct patterns of stress and strain in structures like the skull that would have resulted from various simulated behaviours, such as those encountered during feeding. And what work on Allosaurus, as well as some other work on Carnotaurus, has demonstrated is that although bite forces in these species were relatively weak for their size, the skull was very well constructed to resist powerful vertical forces imposed on the teeth. Now, this, coupled with the very strong jaw muscles reconstructed for these species, suggests that they would not have overcame their prey through delivery of a devastatingly powerful bite, as Tyrannosaurids did, but instead they would have used strong vertical motions of the head and shallow bites to rake the blade-like teeth through their prey, inflicting shallow wounds and gradually bleeding their prey out until it was too weak to fight back. And so, as we can see here that we have very different styles of hunting between different kinds of large theropods, and we could begin to speculate how this may have suited them towards different kinds of prey. So, for example, the very strong bites of Tyrannosaurids may have helped them to brawl with armoured prey of a similar size to themselves, whereas this more attrition, whittling down approach of Carnosaurs may have helped them to tackle relatively large prey, such as juvenile sauropods. And we can also use techniques to investigate herbivorous dinosaurs, calculating bite forces, looking at how they would have fragmented plant stems, and also seeing how they would have accommodated stress and strain in the skull during feeding. And so what this shows us is even in the superficially simple skulls of stegosaurids, therizinosaurs, and sauropodomorphs, we in fact see a great range of functional performance. And what this shows us is that each of these was in fact a very sophisticated feeding apparatus, and that we see each of these groups of animals adapting to herbivory in different ways, and this in turn would have formed the basis of ecological separation between these different animals. And we can also use these techniques to study how dinosaurs moved, supplementing data from fossil trackways with biomechanical models of the musculoskeletal system. So this is some modelling done by workers at the University of Manchester on Tyrannosaurus rex, where they concluded that it is unlikely that adult Tyrannosaurus could have performed a running gait, as this would have placed too much stress on its skeleton. And certainly they would not have been able to chase a jeep at 50 km per hour. Nonetheless, although these higher speed estimates are out of the range of Tyrannosaurus, its power walk would still have enabled it to move at relatively appreciable speeds, perhaps up to around 27 km per hour. Now, if we look at some speed estimates from other dinosaurs, we can see that their real champions are, unsurprisingly, the ostrich dinosaurs like Struthiomimus, that seem to have been able to run at 60 km per hour, similar to living ostriches. So as we've seen, we can use these techniques to reconstruct dinosaur paleobiology in great detail. But what this also allows us to do more broadly is to look at some key transitions in the fossil record, such as exploring the evolution of the sensory structures and brains of birds, as well as the complex evolution of flight within theropoda. What this also allows us to do is to take data from dinosaurs and use it to address fundamental evolutionary questions, such as some of my own work, which has looked at the repeated evolution of herbivory within dinosaurs and use it to investigate the pattern and process of convergent and parallel evolution within the group. So to summarise, if we want to understand the lives of dinosaurs, what we need is not eccentric billionaires plus genetics, but instead careful paleontological work plus maths. Listening. Again, I'm Dr. David Button, currently based at the University of Bristol, and I'd love to hear from any of you. Probably the best way to do it is on Twitter, though I should warn you in advance, I'm very rarely on Twitter, but I'm trying to get better at it, so hopefully do that, then I will get back to you. But again, thanks for listening, and goodbye.